Hello, everyone. Welcome to Creative Biolabs live webinar series. I would like firstly introduce Dr. Sarah Vogelen, who will briefly present our company's products for recombinant antibodies.
Thank you, Kathy, and welcome everybody. So let me first start with a brief introduction of our company and some of the products before we move into the main webinar. So Creative Biolabs, for those of you who don't know us yet, so we have been offering products and services for almost 20 years now in the recombinant antibody field and also related fields. We are located in Shirley, New York, on Long Island. And we provide antibodies in many different formats and categories, such as, for example, research-grade biosimilar antibodies. We have neutralizing blocking antibodies, ADCC, CDC-enhanced antibodies, also anti-drug antibodies and agonistic antibodies. And we offer them in many different types of formats. Here are a few examples listed: IgG, Fab, scFv, also VHH. And in addition, we also offer other products such as mRNA-encoded antibodies, MHC tetramer, soluble T-cell receptor, immunotoxin, and so forth. In addition to our products, we also offer antibody production and development services. We have listed a few examples here. For example, we can do custom antibody production service in case you have, for example, the sequence of your antibody, but no way to produce it. We can produce it for you, or we can also help with non-IgG, IgA, IgM antibody production service, and also produce higher amounts of antibody. For example, we have high throughput recombinant antibody production service in case you have many antibodies to produce. Or we have antibody stable cell line development service in case you need large amounts of your antibody. In addition, we also offer soluble T-cell receptor production service and antibody biosimilar development services. And then moving on, we also have, as a CRO, we have established a broad network of partners of CDMOs who we can activate and connect you with when you're ready to move your project forward into the next stage. And these can help with a variety of different types of solutions. For example, antibody, plasmid and antibody CDMO solutions. We have also partners that can do non-GMP, but also GMP manufacturing. And also transfer, for example, of cell lines. When we generate a cell line for you for antibody production, we can transfer this to a CDMO for larger scale production and process optimization. We can also connect you with CDMOs that have regulatory support solutions, QC, like quality system and inspection management solutions, and supply chain system solutions. And finally, I would like to highlight a few of our featured products and custom production services. The first one is the ADCC enhanced antibodies, which we can help you with, as well as bike and trike production, which I think will be highlighted more in the next few slides when our speaker, Dr. Bruce Walcheck, will talk about this. And of course, we can also assist with bispecific antibodies. We have a few standard formats, but can also do more customized and specific requests. We can help with that as well, customized design. And with that, I would like to hand the floor back to Kathy, who will now introduce our speaker.
Yeah, thanks, Sarah. In this part of a session, we are excited to have Dr. Bruce Walcheck walk us through the targeted Adenine-17 blocker and killer. We will dive into how this innovative strategy cleverly uses Adenine-17's unique dual role to create a powerful immunotherapy approach with great potential for cancer treatment. Now let me introduce today's speaker, Dr. Bruce Walcheck. He is a professor of immunology at the University of Minnesota and has been continuously funded by the NIH for over 20 years. His research focuses on enhancing the anti-tumor activity of NK cells. Adenine-17, a membrane-associated metalloprotease, acts as a kind of brake on NK cells and is often overexpressed in cancers. And Dr. Walcheck's team has developed a unique multi-engager complex that it selectively blocks Adenine-17 on NK cells while simultaneously directing these cells to Adenine-17 on tumor cells. And this will result in a stronger NK cells activation, more proliferation, and better tumor cell killing. And before Dr. Walcheck starts, a quick reminder, and we will have a Q&A session at the end. So if you have any questions during the talk, please type your question into the Q&A panel, and Dr. Walcheck will answer them once he is finished. So now please join me in welcome Dr. Bruce Walcheck. Dr. Walcheck, thank you for sharing your important works with us today, and you may begin whenever you are ready.
Okay, thank you. And assume you can see the slides.
Yes, we can see the slides. If you want to start the pointer, maybe you want to still activate that. Yes, there you go.
Okay, good. I think I'm ready to go. So again, thanks for the introduction. Again, my name is Bruce Walcheck. I'm a faculty member here at the University of Minnesota. And I was going to talk today about natural killer cells, a leukocyte population that we've been focused on, which are capable of killing tumor cells. And in our focus, in part, involves trying to enhance this capability by NK cells. And doing so by targeting a regulatory checkpoint on NK cells called ADAM17, which we initially started to do with a monoclonal antibody. And this antibody has evolved into a multi-engager complex that has various functional effects on the NK cells. And I was going to talk about that today. So I'm sure everyone is probably not overly familiar with natural killer cells. So just a quick overview of these cells. They are part of our lymphocyte populations. So they're derived from a lymphoid progenitor cell, so similar to T cells and B cells. But NK cells are part of the innate immunity in that they don't express a T cell receptor, they don't make antibodies. Instead, they use an array of receptors on their surface to interrogate cells within the body. And so that makes them more similar to these myeloid innate immune cells, which use various pathogen recognition receptors to recognize abnormal cells, pathogens, and foreign material in the body. And so, as I mentioned, NK cells express an array of receptors, and they fall into two categories. They're either inhibitory receptors or activating receptors. And these inhibitory receptors, if they are engaged, as the name implies, that turns the NK cells off and they remain in a resting state. If these activating receptors are engaged, that can activate the NK cell. And it's really a balance between these two, which determines whether the NK cell gets activated or not. When it does get activated, it has two key effector functions. One is to release these cytolytic factors like perforin and granzyme, which can directly kill the target cells or the tumor cells that they're interacting with. And they release various pro-inflammatory cytokines as well that can act on the tumor cells or act on other immune cells. And they release various chemokines that can attract myeloid and lymphoid leukocytes. So they serve as sort of a key bridge between the innate immune system and the adaptive immune system. And one key activating receptor that they express is called CD16A, which is an FC receptor, and I'm going to talk about that quite a bit today. So NK cells can be triggered in various ways to kill tumor cells. Again, their role is to interrogate cells in the body to determine whether they're healthy or not. And healthy cells express class 1, MHC class 1 molecules and low levels of particular stimulatory ligands. Class 1 molecules are engaged by these inhibitory receptors, and that's turning the NK cell off. If a tumor cell, for example, was to downregulate particular MHC class 1 molecules, the inhibitory receptors are not being engaged, and we aren't getting these inhibitory signals, and the NK cells can then be activated by these activating receptors and in turn kill the tumor cell. Tumor cells can may retain their class 1 molecules, but upregulate these stimulatory ligands, such as particular damage-associated molecular patterns. These are recognized by activating receptors on the NK cell that tips the balance towards activation, and then they kill the tumor cell. And another killing mechanism they have is they can recognize antibodies attached to tumor cells. So they have this indirect antigen detection capability. These antibodies, when bound to the tumor cell, can be recognized by Fc receptors on the NK cell, such as CD16A. This is a potent activating receptor. It induces the release, again, of these cytolytic factors that can kill the tumor cell. This process is called antibody-dependent cell-mediated or cellular cytotoxicity. ADCC, and for human NK cells, this is exclusively mediated by the Fc receptor CD16A. So NK cells have received growing attention because of their tumor killing capabilities and attempts to use these as cancer immunotherapies. And they have particular advantages over T cells. Again, they can be activated by this variety of ligands and antigens on tumor cells. And their effector functions are very rapidly induced, unlike T cells that have to undergo a pre-stimulation process and clonal expansion. So their activity takes longer to induce. Because of this broad activity, they can recognize multiple targets across diverse and heterogeneous tumor types. And unlike autologous or allogeneic T cell therapies, NK cell therapies induce lower levels of graft versus host disease because they don't express a T cell receptor, and lower levels of cytokine response syndrome and neurotoxicity than T cell therapies do, as shown in various clinical trials. But NK cells certainly have their limitations as well. Upon adoptively transferring NK cells into a patient, their half-life is shorter than a T cell. So increasing this persistence of adoptively transferred NK cells in patients has been a major area of focus. And there's various regulatory checkpoints that suppress NK cell activity as well. I was going to focus on this protease called ADAM17. It's referred to as an disintegrin and metalloprotease. And 17 means it's just a member of a larger family. What this protease does is it's a transmembrane protein. It sits on the cell surface of NK cells and other cells in an inactive form. But when the NK cell gets activated, ADAM17 literally acts like a pair of scissors. It clips particular receptors and other moieties on the cell surface. And it does it typically at a very specific location, releasing the ectodomain of the substrate and leaving a membrane fragment. And so ADAM17 has been shown to have a very large number of substrates involved in various cellular processes. I would say a lot of these substrates are probably more induced in vitro under very specific conditions. So the physiological relevance of some of these as ADAM17 substrates is not real clear. But there are a number of very well characterized ADAM17 substrates, one being CD16, and L-selectin or CD62L. I'll talk about that a little bit as well. So we had previously shown a number of years ago that ADAM17 cleaves CD16 on NK cells and acts as this negative feedback loop. So when CD16 engages an antibody on a tumor cell, this activates the NK cell, it activates ADAM17, and it cleaves CD16A. And this likely functions as a homeostatic process to regulate the cell surface density of CD16 on NK cells, because it's a potent activating receptor, and these are very highly cytolytic and pro-inflammatory cells. So this is a way of quickly regulating their activity. However, if we get excessive shedding of CD16A, that's certainly going to reduce the ADCC potency of NK cells and limit their ability to kill these antibody opsonized tumor cells. And so we've been focused on trying to disrupt this process to preserve CD16 on the surface of these cells. One way we've done this is just by characterizing the cleavage site of CD16, engineering a mutation there, and generating a non-cleavable version of CD16 that we can express in engineered NK cells. And these type of NK cells are being tested in clinical trials now. But this is an expensive and labor-intensive process to engineer NK cells, manufacture and expand them. These are going to be allogeneic NK cells as sort of a off-the-shelf therapy. So that means the patient's going to have to be conditioned by depleting their lymphocytes, so we get less of this host versus graft rejection. So again, an expensive, labor-intensive process. We were interested in other approaches to prevent CD16 cleavage that could then be used for endogenous NK cells in patients as well as adoptively transferred NK cells. And so we've been utilizing this antibody we call MEDI1 that selectively binds to human Atom-17 and blocks its function. And so we've shown this in a number of ways, but here in this sort of straightforward assay, we just activate enriched NK cells with PMA. This is just a 4-ball ester. It overtly activates NK cells and induces Atom-17 activity. And then we just stain for CD16 by flow cytometry. So on resting NK cells, we find there's the majority of NK cells have CD16. We activate them. We get this appreciable downregulation in CD16 expression. If we activate in the presence of MEDI1, we block this downregulation of CD16. And TAB16 does that as well, but I'll get into what that is in a few minutes. So MEDI1, it's a human IgG antibody, and our initial interest with it was just to block Atom-17 to retain CD16 on NK cells so they could mediate better ADCC. But we found that actually the FC region of MEDI has some very interesting effects on NK cells as well. And so I was going to show some of that data here. So one of the assays we did is we just take enriched NK cells that we isolate from the peripheral blood of our donors, and we stimulate them with IL-15, interleukin-15. This is a cytokine that's very commonly used to stimulate NK cells. It primes them. It induces their proliferation. It's used in patients to stimulate endogenous NK cells and to increase the persistence of adoptively transferred NK cells. And so here in this assay, we're just taking NK cells or NK cells in the presence of MEDI and then activating them with IL-15. And we're just looking initially in this assay at the activation of the NK cells. And here we're looking at these very early activation markers that get upregulated on NK cells, such as CD69 or CD25, which is the IL-2 receptor alpha, or CD137, which is also referred to as 41BB. So this is a bit of a busy slide, but what we're doing here is we're just treating NK cells, unstimulating them, activating them with IL-15 or treating them with IL-15 and MEDI. We're looking at the expression level of these different markers that I mentioned, and we're looking at a time frame from day 1 to day 7. So on unstimulated NK cells shown in these blue histograms, they express very low levels of CD69, CD25, and CD137. For CD69, when you activate NKs with IL-15, it upregulates very rapidly, and it remains on the cell surface over this time frame that we're looking at. If you have MEDI along with IL-15, essentially see the same expression pattern. However, if we were to look earlier than 24 hours, we do find that MEDI increases the rate of CD16-9 expression, but otherwise its expression pattern looks pretty similar. However, CD25 and CD37 are considerably different. Again, very low levels on resting cells, activate them with IL-15. Over time, we get some upregulation of CD25, and then it diminishes by day 7. But in the presence of MEDI1, we see this very drastic upregulation of CD25, though, again, it diminishes over time. And the same with CD137, this very overt upregulation of CD137. That's transient and by day 7 has returned to normal levels. Here, we're just quantifying the data, looking at NK cells from a number of different individuals. And I wasn't showing the data here, but if we just treat NK cells with MEDI alone, we do not get activation of these cells. The expression pattern of these markers looks like it does by these unstimulated NK cells. So we also looked at the proliferation of NK cells under these conditions. And to do that assay, we label our NK cells with a cell dye called cell trace, and results in very bright fluorescent labeling of our cells. And as they die, this fluorescent label is going to dilute out. So it allows us to quantify the level of proliferation and the number of rounds of division by these cells. So that's shown here. So here we have our labeled NK cells. They're unstimulated. We're looking at a time frame from, again, 1 to 7 days. These cells, again, this is done by flow cytometry, and these cells remain very fluorescently bright over the 7 days because they're not diluting out this dye, because they're not proliferating. With IL-15, though, however, we start to get some proliferation of our cells, and you can see these different rounds of division that the NK cells are undergoing by day 7. If we treat with just MEDI alone, not much of an effect on cell proliferation, but MEDI with IL-15, we see this dramatic increase in NK cell proliferation. And again, that's just quantified over here using NK cells from a number of different donors. And we see this in vivo as well. If we just take an immunocompromised mouse, inject human NK cells into them, and then also administer IL-15, shown by these blue dots, at various time points up to day 21. And in a group of these mice, we also administered our MEDI1 antibody at these different time points as well. And then at these different time points, we just draw blood from these animals and quantify the human NK cell numbers in circulation. So if we look at our mice that were treated with IL-15 only, the NK cell numbers will increase over time because they're proliferating due to this IL-15. If we just look at day 2, we find that mice treated with IL-15 or IL-15 with MEDI, the NK cell numbers are the same, indicating that we are administering the same number of NK cells. But over time, in these mice that received IL-15 and MEDI1, we see this dramatic increase in proliferation of these cells. So again, in vivo, we're seeing this enhanced proliferation of NK cells as well by MEDI1. So we're curious what is happening. We're stimulating ourselves with IL-15, and MEDI1 is enhancing their activation and their proliferation. So we're getting some sort of synergy between MEDI1 treatment and IL-15. So it could be that we are blocking the shedding of a particular substrate, and that's inducing this signaling, resulting in this synergistic effect. However, MEDI1 is a human IgG1 antibody. And so it's possible that it could bind to Atom-17 and actually be recognized by CD16. And so CD16 is mediating the shedding. And MEDI1 would also be blocking Atom-17, preventing CD16 shedding, so prolonging the signaling as well. So to address this, we just made various versions of MEDI1 where we swapped out its FC region with different isotypes like IgG4. IgG4 is known to bind CD16 with at least 10-fold lower affinity than an IgG1. We engineered these mutations into the cytoplasmic tail that are known to prevent IgG1 antibodies from binding to CD16, or we just removed the FC region in this antibody. So all of these antibodies equivalently block Atom-17 function. I'm not going to show that data here. And so we looked at what the effects of these different antibodies had on NK cells in terms of the activation and the proliferation assays that I showed you. So we treated with these individual antibodies in the presence of IL-15 and looked at these readouts. And here I'm just showing the cumulative data. But looking at the upregulation of CD25 and CD137, we find that IL-15 alone upregulates these a little bit. Dramatically increased if we have IL-15 and MEDI1, but significantly decreased when we combine IL-15 with either of our MEDI1 variants. Same case with CD137 expression. And the same thing with if we look at proliferation of our NK cells. IL-15 increases proliferation somewhat, appreciably increased if we have IL-15 and MEDI1, and significantly decreased compared to MEDI1 when we treat with our antibody variants instead. So we feel this data and a bunch of data I'm not showing that is reported in this paper was consistent with his model that MEDI1 can block Atom-17, but to be engaged by CD16. CD16 signals that synergizes with IL-15 signaling, enhancing the activation of these cells and inducing the upregulation of markers like CD137. And this leads to increased NK cell proliferation. In data I wasn't showing for the sake of time, though, to get this increased proliferation, we do have to have accessory cells in there that express CD137 ligand. So CD137 is a well-characterized co-stimulatory receptor that can drive NK cell and T cell proliferation. And we find that in the presence of cells that express its ligand, that is necessary to drive this increased proliferation. We can also leverage this increased expression of CD137 and CD25 using feeder cells that express high levels of recombinant CD137, and then using the cytokine IL-2. So we find that if we were to pre-stimulate our NK cells with IL-15, co-culture with our feeder cells in the presence of IL-2, we get an appreciable expansion of our NK cells. However, if we were to pre-treat these cells with IL-15 and MEDI and then perform this proliferation assay, it's significantly increased in this case, so we get a much higher level of proliferation. And we think this might be something useful for the ex vivo expansion of NK cells. These types of feeders are commonly used to expand NK cells for adoptive transfer in patients. And so this approach of using IL-15 with MEDI may be a way of increasing NK cell proliferation, generating more NK cells for adoptive transfer, or requiring fewer rounds of this process. However, MEDI does have its limitations. It's going to block ADAM17 on cells other than NK cells, and most cells in the body do express ADAM17. And as I mentioned, ADAM17 has a lot of substrates too involved in these various cellular processes. So it could be potentially detrimental to administer ADAM17 to patients because of all the other substrates of that protease that will be affected. And another downside of CD16A in regards to MEDI1 is that there are different allelic variants of CD16A that bind to IgG with low or high affinity. So they're going to bind to MEDI1 with low or high affinity. And it's possible this is going to lead to different levels of NK signaling by MEDI1. And so this is the effect of MEDI1 is going to vary between individuals because of these allelic variants of CD16A. So to address this limitation, we made this bispecific antibody that consists of an scFv from MEDI1 and a humanized camelid anti-CD16 antibody. So this is a single domain antibody. And it has some advantages using it in this complex because it's a smaller version of an antibody fragment, and it potentially reduces steric hindrance with the other components that we may be attaching to CAM16, such as the MEDI1 scFv. So we call this complex TAB16 or targeted ADAM17 blocker because we're attempting to selectively block ADAM17 in NK cells that express CD16. But we expect it to potentially work like MEDI1, which is engaged by CD16A and blocks the function of ADAM17. So to check out its potential selective activity, reactivity with NK cells, we have a version that's been tagged with a 10x hist tag, and we can detect that with a antihistidine antibody and a second stage antibody. So here we just treat mononuclear cells with our TAB16, and we can detect it staining with this anti-his antibody. So if we just stain human mononuclear cells with a second stage antibody, very low levels of staining, stain them with an anti-his second stage antibody, very low levels of staining. Or if we stain our mononuclear cells with our TAB16 plus our anti-his in second stage, we find very little staining of T cells, but a very appreciable staining of NK cells. And again, this is just quantified over here using NK cells from different donors. So NK cells can be identified by the expression of CD56 and the lack of CD3, which is a T cell marker. And CD56 is expressed at different levels by NK cells circulating in the blood. They're either CD56 dim or CD56 bright. The CD56 dims are the mature NKs, and the brights are referred to as immature NK cells. And the mature NK cells express CD16. So here we're showing CD16 staining of the CD56 dim cells. If we look at TAB16 versus 56 instead, we find that TAB16 is binding to these 56 dim cells, but not the CD56 bright cells. And again, these are the cells that lack CD16. And we find that, again, CD16 is not binding to T cells. This blue contour is just NK cells stained with our control antibodies. So we're finding that TAB16 is selectively binding to NKs and to NK cells that express CD16. Now we wanted to test the MEDI scFv component of our bispecific antibody to make sure it's still blocking Atom-17. So here we're just doing our NK cell activation assay with PMA. Here, the gray histogram is resting NK cells. They have high levels of CD16. They downregulate CD16 upon activation. MEDI1 is blocking that. And we find that TAB16 blocks that as well. And again, that's quantified over here using NK cells from a number of donors. And we find that TAB16 is blocking CD16 downregulation as efficiently as MEDI1 is, shown in the blue versus the red bar graphs. CD16 is not expressed on T cells, and we wanted to see whether this blocking effect by TAB16 was specific to NK cells versus T cells in our mononuclear population. So we looked at another substrate of Atom-17, that is L-selectin or CD62L. Here, a subset of NK cells, resting NK cells, express L-selectin. You activate them with PMA. It's very efficiently downregulated. MEDI1 blocks that, and we find that TAB16 blocks that as well. We have a larger subset of T cells express L-selectin. Again, it's very efficiently downregulated, and when you activate them with PMA, it's blocked by MEDI, but not as efficiently blocked by TAB16. So TAB16 efficiently blocks L-selectin on NK cells, but not as well on T cells. So this is showing that MEDI1 can block Atom-17 on both these leukocyte populations, but TAB16, by engaging CD16, is selectively blocking Atom-17 on NK cells. Even though T cells express Atom-17, they don't have CD16, and we're not seeing inhibition of Atom-17 in those cells. So I showed that treating NK cells with MEDI1 and IL-15 will increase their activation and their proliferation. And we're curious whether TAB16 in combination with IL-15 does the same thing. So here we're just treating NK cells with IL-15 or MEDI1 or TAB16 and looking at their upregulation of activation markers. Here I'm just showing CD137. And this is using NK cells that have been stimulated in this manner for 24 hours. So unstimulated cells, again, express very low levels of CD137. Even stimulated cells by 24 hours are expressing very low levels of CD137. Dramatically increased in expression by MEDI, as I've shown, and we see the same thing with TAB16. I'm not showing the data here, but if we just use TAB16 by itself, it does not induce this upregulation. It has to be a combination of IL-15 and TAB16 that does that. Again, we looked at NK cell proliferation too, using here just our dye dilution assay. We find that unstimulated NK cells are not diluting out the dye. They're not proliferating. With IL-15, we're getting some proliferation of NK cells. With MEDI alone, at this dose of 33 nanomolars, we're not getting any proliferation of our NK cells. But if we combine IL-15 and MEDI at this concentration, we see this enhanced proliferation of NK cells, as I showed previously. And we see the same thing if we were to combine IL-15 with TAB16 as well. And this effect dilutes out as we reduce our concentration of TAB16. And this data is just quantified over here on this side. And again, if we were just to treat NK cells with TAB16 alone, we are not inducing their proliferation as occurs when we combine it with IL-15. So we're also curious what happens when you treat NK cells with TAB16 and co-culture them with tumor cells in terms of their capability of killing those tumor cells. So the tumor cell here I'm using in this assay are SCOV3 cells. This is a human ovarian cancer cell line. And so we co-culture enriched NK cells with our tumor cell line in the presence of IL-15 over a period of a couple of days. Our tumor cell has been transduced to express a viability marker like nunc light green or nunc light red. And as these cells die, they lose that fluorescent color. We can also look at their death by apoptosis as well, but I'll just be showing the loss of nunc light red. So here we just treat co-culture NK cells with tumor cells in the presence of IL-15. Do the same thing, but we add Herceptin to this assay. This is an antibody, a human IgG1 antibody that can recognize HER2 on the ovarian cancer cells. So we're going to induce ADCC in this assay. Or we co-culture NK cells, tumor cells, IL-15 in the presence of TAB16. And so here I'm showing viable SCOV3 cells over time. We're doing this assay out to 72 hours, and we're just doing some live cell imaging over this time frame. And all this data has been normalized, double normalized to the SCOV3 untreated cells. So SCOV3 cells really remain at 100% in this assay. If we co-culture them with NK cells, as shown in the blue line, we get some killing of our SCOV cells through natural cytotoxicity. If we add Herceptin to this assay to induce ADCC, we get an appreciable increase in killing. And interestingly, if we add TAB16 instead, we get an equivalent level of killing as if we had Herceptin in there. And that's just quantified over here in the bar graph. So it's possible that when TAB16 engages CD16 and ADAM17 on the NK cells, induces cell signaling that synergizes with IL-15. We're just enhancing the effector activity of these cells, and they kill tumor cells better. But it is known that ovarian cancer cells express very high levels of ADAM17, and it's actually associated with increased levels of metastases and poor patient outcome. And so it's conceivable that maybe our TAB16 is also linking CD16 to ADAM17 on our tumor cells and inducing ADCC. So to test for that, we CRISPR knocked out ADAM17 from our SCOV cells to remove it and prevent ADCC from occurring when we add TAB16 to our cells. And here I'm just showing that we effectively remove ADAM17 from our cells. I'm staining wild-type SCOV cells or ADAM17 knockout SCOV cells. And for the wild-type cells, we see that they express high levels of ADAM17 and much lower levels in our knockout cells. If we were to stain for HER2 instead, its levels remain the same in our wild type and our knockout cells. And so if we were to do our killing assay again, we find that NK cells co-cultured with wild-type SCOV cells in the presence of TAB16 and IL-15, the green line, mediate very efficient SCOV cell killing. But the same assay using our SCOV3 knockout cells, we get a greatly reduced level of killing that occurs at this natural cytotoxicity level. And again, that's quantified over here just using different effector to target ratios where we have 2 NK cells to 1 tumor cell, or we have 1 NK cell to 2 tumor cells. Again, a significant difference in killing if our SCOV cells have ADAM17 or lack ADAM17 by our TAB16 molecule. So we feel this data I'm not showing is consistent with TAB16 binding to CD16 on NK cells, selectively blocking ADAM17 inducing CD16 signaling that synergizes with IL-15, enhances the upregulation of coactivation receptors like CD137 that can be leveraged to increase their proliferation, but it also links CD16 to ADAM17 on tumor cells. However, there's still some limitations of using our TAB16 molecule and IL-15 if, for example, down the road this was to be delivered to patients. We would have to produce two different compounds, and so we're going to have this lot variability between these two different compounds, and we're going to have, by administering two different compounds to patients, pharmacokinetic differences between them. So to address this, what we did is we made another complex where we linked our CAM anti-CD16 to IL-15 to our MEDI-SCFV. And really just to kind of make a longer story short, we find that this complex also selectively binds to NK cells. It links NK cells to tumor cells that express ADAM17 better than tumor cells that lack ADAM17, and it drives increased proliferation in the absence of exogenous IL-15. And so with this complex, we refer to it as having a dual function in that, again, it engages CD16 on NK cells, selectively blocks ADAM17, induces NK cell proliferation, but also can engage ADCC on tumor cells. But since time is getting short, I just wanted to end with really our last iteration of this multi-engager complex that we've been working on, with a few more improvements that we've made. So the IL-15 receptor itself consists of three components. It has a beta component and a gamma component. That's expressed in the NK cell. And then there's an alpha component that binds the IL-15 and presents it to the beta and gamma chains. IL-15 can bind to beta and gamma on its own, but it occurs much more efficiently when it's associated with the IL-15 receptor alpha component, which is normally expressed by monocytes and dendritic cells or occurs in a soluble form as well. So we generated another version of MEDI-SCFV where we linked it to the a truncated version of IL-15 receptor alpha. And with this, we can complex it to CAM16, anti-CD16, and IL-15. So it really functions like the TAB16-15 molecule I just talked about, but it presents IL-15 to NK cells much more efficiently due to this IL-15 receptor alpha complex. And another thing we can do with it is we can complex it to these tri-specific killer engagers. So these are molecules that consist of CAM16, IL-15, and another SCFV or camelid antibody that can bind to different tumor antigens. And it's well characterized that these trikes enhance NK cell killing of tumor cells in preclinical trials. And some of these are being tested in clinical trials or in pipelines to be tested as well. And with our MEDI-SCFV IL-15 receptor module, we can complex to these trikes, so we enhance NK cell function and can engage another tumor antigen. So we're just at early stages of looking at this complex, but this is something we feel that has advantages of enhancing NK cell priming and proliferation by mechanisms that I talked about, but also could engage ADAM17 when it overexpressed on tumor cells, or can engage another tumor antigen on the tumor cells, allowing us to target more than one tumor antigen. And this is important as a means of trying to reduce or prevent tumor variant escape, which occurs with CAR T cells and monoclonal antibodies that target only one tumor antigen. You apply this immune pressure to these tumors. This can result in tumor cell antigen variants that lack that antigen, and they continue to grow, resulting in resistance and relapse by these tumors. But by being able to target more than one tumor antigen, we can reduce the likelihood of this tumor escape from occurring. So I think I'm at time, so I'm just going to end with this. Just want to quickly acknowledge some people in our lab, current members and previous members, and some of our key collaborators as well. In particular, this Martine Felicis was this immense expertise in generating these multiplexed antibodies, and the ones I talked about today have been generated by him. So he's been a key collaborator. He's part of Jeff Miller's larger research lab, and some of other members in his lab have helped us, as well as collaborations with this Jeff Hart. So with that, I'll end it and see what kind of questions we have.
Great. Thank you so much, Dr. Walcheck. Let me start to share here. Yes. Okay. Yes. So thank you so much. That was a very insightful presentation, very exciting. I hope our audience found it as engaging as I did. So now, without further delay, let's move on to our Q&A session. Just a quick reminder for the people attending, please type your questions in the chat, and then we will handle them. So I see there are already a few questions here, so let's start with those. So first of all, Dr. Walcheck, the M1 and M2 macrophages in the tumor microenvironment may express CD16. Does TAB16 bind to macrophages, and what is the expression level of ADAM17 on macrophages?
Sure, yeah, it's an interesting question. There are a subset of peripheral blood monocytes that express CD16. It's a very small subset. And in terms of macrophage, monocytes that are in the tissue, a subset of them can express CD16 as well. And I don't know in terms of sort of myeloid-derived macrophage, whether they express CD16 or not, but they're a key component of the tumor microenvironment. So if they do express CD16 or M1 macrophage express CD16 in the tumor microenvironment, it's certainly possible our TAB16 could engage those as well. And what might occur, we don't know. It might be a way of linking NK cells to these suppressive monocytes in a way of perhaps eliminating those macrophage. By engaging CD16 on those macrophage, it may alter the function of them. There's a lot of different possibilities. It's certainly something that needs to be looked at, and I don't have a great answer for.
Thank you. And moving on to the next question. In vitro assays, we do not see much difference in the effects of MEDI1 and TAB16. Would you expect a drastic difference with TAB16 as compared to MEDI1 in vivo?
Yeah, interesting question. We haven't yet compared like TAB16 to MEDI because we've been generating these newer versions of our multi-complexed antibody, trying to get to a final product that we wanted to more move forward to our in vivo studies. So TAB16 was kind of a transition molecule. But if we were to compare a multi-engager to MEDI1 in vivo, I mean, I think there would be certain similarities that would occur in terms of enhancing the activation in the proliferation of NK cells. I think the TAB16 is going to be much more efficient at linking NK cells to tumor cells if they overexpress ADAM17. So I think that'd be an advantage of the multi-engager complex. And again, MEDI1 could potentially have some more detrimental effects because it's going to block ADAM17 on other cell types and the shedding of other substrates. That could be beneficial. There's certainly examples of I could provide where that might be beneficial, but it could be detrimental as well. We don't know that. So those are just things, that need to be looked at.
Thank you. We have another question. Is there an ADAM17 inhibitor that can increase NK cell proliferation in vitro?
An NK cell, well, we know that our antibody as an inhibitor can increase NK cell proliferation in vitro. There are small molecule complexes that inhibit ADAM17. Their selectivity for ADAM17 is not nearly as good as a monoclonal antibody or our monoclonal antibody. And we've done this a little bit where we really don't see enhanced proliferation of NK cells by our ADAM17 inhibitors because we're not getting CD16 engagement by them, and we're not getting this co-stimulation that occurs as with MEDI1 or multi-antibody engagers. So we really haven't seen it with the limited studies that we've done with small molecule inhibitors that we have.
Okay, thank you. And then maybe more of a general question. If you have NK cells that are producing these factors that will kill the tumor cells, like granzyme and the other one, are you not worried about bystander effects, like bystander cells that might also get these compounds that will then result in killing of the cells that are not intended, like that are not tumor cells?
Yeah, I think that is a issue with any sort of cell therapy is you're going to have perhaps bystander cell killing. So I think that's really unavoidable and may just be a consequence that you have to deal with these type of immunotherapies. Bystander killing could be beneficial too because there are a lot of cells in the tumor microenvironment that help the tumor cell grow. So if we're getting bystander killing of them, I would see that as being beneficial. But bystander killing of other cells is a possibility. And so that's why we're trying to develop multi-engagers that are as selective as possible. But right, when they release these granzyme and perforin compounds, I think there is going to be a zone of killing around these cells, mostly directed to the tumor cell because that's how they release them. But I feel bystander killing will occur to some degree, and I think basically unavoidable too.
Okay, thank you. Thank you so much for explaining that and answering all these questions. I think our time is up, so I will return the floor back to Kathy for the concluding remarks.
Thanks, Sarah. And thank you, Dr. Walcheck, for the insightful presentation today. And please note that Creative Biolabs regularly invites estimated experts in life science and biotechnology to share their latest findings in our webinar series. We encourage you to visit our website and subscribe to our social media channels so you won't miss any upcoming events. So that's it for today. And thank you everyone for joining us. And we will see you next time.